If you've optimized it one time is enough. We can get above 80% delivery and viability pretty easily for most cell types. The things you vary are the pore sizes vary. So for example, a T cell is a lot smaller than a stem cell. So you have a smaller pore size for T cells than you would for stem cells. The other thing you vary and you optimize is the speed of the cells. If you go too slow, it's not going to work. If you go too fast, you're going to kill them. So you need to be in this kind of zone where they're going fast enough for this to work, but not too fast for you to kill them.
Hey there, biotech innovators. Welcome to the Smart Biotech Scientist podcast. Today, we're diving deep into the future of cell therapy with Armon Sharei, who is the founder and CEO of Portal Bio. We'll explore the challenges of intracellular delivery and discover how point-of-care manufacturing could revolutionize cell therapy. Grab your coffee and get ready for some serious biotech inspiration. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brühlmann, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Welcome, Armon. It's great to have you on.
Thanks, David. Good to be here.
Awesome. Armon, share something that you believe about bioprocess development that most people disagree with.
Ooh, fun. I'm very cell therapy biased because that's my background. So I would say I really think point of care cell therapies is the future, meaning that you would produce the cell therapy at the hospital with the machine next to the patient and it just immediately gets reinfused. Many people don't think that'll happen, but I totally think that's the future.
Yeah, fantastic. And we're going to dive a bit more into all that. But before we do that, I'd love to hear from you about you, because your journey from MIT to founding multiple biotech companies is fascinating. What first sparked your interest in cell engineering, and what were some interesting pit stops along the way?
I guess it started with I had just started grad school for my PhD at MIT. I was chemical engineering. And when you're looking across projects to join for a lab, the one that had really caught my attention was one of the professors was developing a gun to shoot materials into cells. And as a 20-something-year-old, they're like, well, that sounds like a cool American solution to a problem. We should do that. But then also, it was interesting because it made me think through the question of like, well, cells are the little machines that make everything happen in the body. And if you can put stuff in them, that's what allows you to control them. And if you could control cells, there's so many things you could do therapeutically. It would be way beyond what a small molecule or a biologic does. So that's what got me interested initially. And so I joined that project. We worked on that gun system. Over time, we accidentally had figured out that you don't need to shoot things. You can just squish them instead. So we had figured out that if you just squish cells at high speeds, you can disrupt the membrane, put whatever you want in them. And so in 2013, when I was graduating, we ended up starting Squeeze, our first company. And that was born out of the fact that once we had figured out this whole squeezing phenomenon, there were a lot of partners that we had started working with on the academic side, and there was a lot of interest in it. So we started the company to just commercialize that. And I actually didn't join it full time. I went and did a postdoc in immunology at Harvard Med School because I initially wanted to be an academic. And then as things continued with Squeeze, I ended up jumping full time. Short version of all the rest was we got a big partnership with Roche a few years after. We raised about 300 million in equity financing over the years, another 100 million from the Roche partnerships. We went public in 2020. And then towards the end of 2022, the kind of short version is me and the board agreed to disagree. I really wanted to open up the technology side more. They really wanted to focus on one of our clinical programs. So from there, we got Portal, our new company, going early last year, which is kind of a next-gen version of what we used to do. But this time, the business model is very different. We don't do therapeutics. It's just instrument and consumables and platform access. So yeah, at this point, we have over 50 partners, including 9 of the big pharma. So it's been moving.
Now, it's a fascinating journey, and you mentioned that you wanted to be an academic. Now you're CEO of a company. That's quite a different role. How do you feel about that? Do you miss the academic world, or finally the business world is a better world?
Yeah, I don't miss it. I'll admit that at the time it was extremely 50-50 whether or not I jumped. Because I think when you're in the academic world, you drink the Kool-Aid of we, the academics, are the smart, higher level beings. We come up with all the crazy new innovations, and then some schmuck in industry can go translate it. That's the easy part. And you kind of buy into that. But then when I eventually jumped, I was like, there's like a lot going on here on the industry side. This is where the rubber meets the road on actually trying to get things out there and make a difference. So I'm glad I did it. I think the parts I probably like the most is that it can be much more team oriented on the industry side, whereas academia, it's inherently about first author, last author. It's very much an individual sport. And I think the other thing is just proximity to the impact is also what I like. I will say there's no ceiling to what you can achieve on the industry side, but there's also no floor, so things can crash and burn completely. In academia, there's both, for better and for worse, but I'm happier in the no floor, no ceilings world, I guess.
Sounds like a big adventure. And now, well, you're moving the needle again with your new company, and intracellular delivery is critical to many research and clinical applications. So what key scientific challenge made you realize we needed better delivery methods?
Yeah, so if you think about a cell as that kind of little machine, there's many different types of materials you would want to put inside it to alter its functions, because that's ultimately how you give these cells instructions, is how I would think about it. And kind of looking at the field of what exists, and we had a review in Nature back in 2016 on these, there's very limited options that work across cell types. Basically, the only thing that works across applications is what's called microinjection, where you literally take a needle and stick it into a cell. So it's extremely unscalable, but that's the only way you can do whatever you want. A lot of the technologies that exist right now, like nanoparticles or electroporation is another technique where you electrocute cells. These systems are very dependent on the charge of the molecule you're trying to deliver. So DNA and RNA are highly negatively charged, and they're chemically pretty uniform. So they work with that. But they have a lot of trouble with other materials like peptides or proteins or impermeable small molecules, et cetera. Delivering multiple things is hard. And then also there's a cell function issue where cells don't like getting electrocuted. So even though maybe you got your material in, the cell is actually really mad at you and isn't behaving normally. So what was the point? You kind of broke the machine while you were trying to put something into it that's not going to serve a purpose a lot of times. So a lot of these challenges kind of have compounded the issues of what you need to do to engineer a cell.
Yeah, bringing material into a cell is definitely a difficult challenge. And you mentioned electroporation. And I remember when I worked in cell line development, even that was a challenge with just DNA. I mean, it's not that simple. So before we look into more detail into your technology, let's talk about the challenges, because yes, you said you want to deliver material into the cell to do all kinds of things. Maybe tell us what kind of material that can you bring into the cell, and what are the fundamental challenges with crossing that barrier?
It varies by kind of what your application is, obviously, but let's say you're trying to put a piece of DNA into a cell for cell line engineering. The way you can think about the barriers is, well, your first barrier is the lipid bilayer to get into the cytoplasm. And then you need to get through the cytoplasm to the nucleus. The nucleus has its own membrane that you need to get through. So there's kind of multiple steps you need to be able to pass through to get there. And this is where something like electroporation, because of the electric field, it essentially fires the DNA into the cell because DNA is highly negatively charged. It gets repelled from the negative electrode and it just fires through the cell. And that's part of how electroporation works. Nanoparticles, they tend to be positively charged. They complex with the DNA. And because they're positively charged, they stick to the negatively charged outer cell membrane that helps get them endocytosed. There's this obscure dance that happens after that seems to let some material get released into the cell, which then subsequently can go into the nucleus and get incorporated. So there's kind of a many-step process. If you take another extreme, like let's say you're trying to put a peptide into a cell, there's almost no real options right now because the charge and function of a peptide is not uniform. It can vary drastically depending on what the peptide is. So where an electroporation or a nanoparticle really require that thing to be highly negatively charged, that's not going to be true most of the time. So they just can't deliver it and you end up with nothing really, or you just hope some of it gets endocytosed into a cell.
And how are you now solving this challenge with the technology you're developing?
The main kind of insight we had from grad school was that if you mechanically deform a cell at high speeds, you can physically disrupt the lipid bilayer, the cell membrane itself, such that now it's kind of open to the surrounding environment and diffusion can just pull things in. And the way we've implemented this at Portal is we essentially have what looks like a fancy filter. So it's a thin piece of silicon with holes in it. The holes are designed to be slightly smaller than the cells. So for example, if you have a 7 micron T cell, it's going to go through a 5 micron hole. So as the cell is transiting through these holes, if you run them fast enough, the rate at which the cell needs to deform becomes too fast for the lipid bilayer, and so it just tears. Think of it as you had a balloon filled with water and just like a bunch of tears occurred. It doesn't pop, but just a bunch of tears occur. Now you can just have material diffusing in through those tears, and the cell takes about 30 seconds to close them back up because the lipid bilayer is ultimately pretty fluid. It'll just seal it back up. But during that window, whatever's outside can go in, and that's why in our case, we're pretty independent of material or even cell type in getting this to work because it doesn't matter if it's a piece of DNA out there or a peptide or a small molecule or a piece of RNA. They're all just going to diffuse into the cell when the membrane is torn.
And how efficient is that process? Do you have to repeat that several times or these 30 seconds are okay?
No, if you've optimized it one time is enough. We can get above 80% delivery and viability pretty easily for most cell types. The things you vary are the pore sizes vary. So for example, a T cell is a lot smaller than a stem cell. So you have a smaller pore size for T cells than you would for stem cells. The other thing you vary and you optimize is the speed of the cells. If you go too slow, it's not going to work. If you go too fast, you're going to kill them. So you need to be in this kind of zone where they're going fast enough for this to work, but not too fast for you to kill them.
Okay, but bringing the material into the cell is one thing, but how do you ensure that actually it's used? Finally, you have gene expression or you want to change whatever metabolism or what? How does that work?
So that part becomes pretty dependent on the material and what biology you're after. Luckily, most things should either be directly functional in the cytoplasm or have a way of going from the cytoplasm to their target location. For example, RNA works in the cytoplasm. You deliver a piece of mRNA or circular RNA or what have you. The ribosomes are in the cytoplasm. They're going to latch onto it, start translating it. That works perfectly fine. If you're doing something like CRISPR complex, which needs to go edit the DNA, the CRISPR protein, along with its guide RNA, the CRISPR protein actually has what's called a nuclear localization sequence on it. So if you put it in the cytoplasm, there's transport mechanisms that grab onto that sequence and pull it into the nucleus where it can then go and edit its DNA. One of our weak points is that if you put a plasmid into the cytoplasm, it doesn't have its own way of going to the nucleus. It just kind of stays there. And that's an example of one where you would need to put in some additional chaperone protein or something to go pull that plasmid in. So that's one thing we have to account for depending on the application is your cargo going to be directly active in the cytoplasm, or is it going to have its own way of doing what it needs to do? Or do you need to assist it in some way by co-delivering something else?
Okay, I see. Yeah. And I guess there's a lot of optimization involved in that, depending on your molecule.
If you're replicating normal biology, there's usually not much optimization. So for example, RNA is meant to be in the cytoplasm. Honestly, you don't need to optimize anything. It'll just work. CRISPR complexes have a nuclear localization sequence. They'll just go there. Or if a peptide was supposed to block some function in the cytoplasm, it'll just do it. The DNA thing is more of an uphill battle. You would have to optimize that because now you're throwing in a chaperone to trick the cell to take the DNA into the nucleus because DNA isn't meant to be in the cytoplasm. So there's different complexities there that can come up.
What are currently the most challenging proteins or molecules to bring into a cell?
I would add in the layer of complexity of it depends on the cell type. So for example, primary cells like T cells or stem cells or whatever from you and me, even things like RNA and DNA can be very difficult to bring in there. So I would say one of our biggest use cases, people trying to put things like RNA into immune cells. Because there's a lot of potential therapeutics where if they took your immune cells out from your blood and you put in an RNA, you can engineer them and go after a particular disease or function. Then there's certain cargoes that are almost universally difficult. And that would be things like peptides or proteins, also a lot of impermeable small molecules, which gets into this zone of we've had a lot of uptake for drug screening. Because when big pharma, for example, is screening for drugs or these biotech, they might have a target of interest. They will do a biochemical assay with 1 million or billion compounds. They'll find, let's say, 10 hits. But many of these hits might actually be impermeable to a cell, especially the newer generation stuff they do, like protacs and peptides, they tend to be impermeable. And so they face this interesting question of, I have 10 hits, they're impermeable to my cell. Which one am I going to spend 2 years doing chemistry on to make it permeable? Because then I might find out it was actually never going to work because some biology wasn't going to happen. And in our case, they put it as skip to the answer. Because if you have 10 hits, we can just deliver all 10. You'll see which one would have actually worked. And then you can only go invest in doing the chemistry on that one.
How exactly does your technology work for drug screening?
So it's the same principle of you, in this case, would have your cell type of interest that you're using for your screen. And then you would run it through our membrane. You could have either mixed that impermeable drug with it pre-mixed. So now the cells plus the drug are going through. When the cell opens up, the drug just diffuses in. When you really want to go to high throughput screening, we kind of exploit a different aspect of this, which is, like I mentioned, the cell membrane stays open for 30 seconds. So what you can do is you can have many 96 well, 384 well plates with the drugs of interest in each well. And you can just run cells through our membrane without any cargo. And once they get disrupted, as long as you dispense them within 30 seconds, they will still take up whatever's in the well. So that's kind of a simple trick to go high throughput. You don't need to mix every time for every little well. You just have a dispenser that's dispensing disrupted cells, and they'll just take up whatever's in the well.
That's actually a rather simple way to all that screening.
Yeah.
Obviously, the big pharma, they have robotics, so you can screen tons of different drugs using that technology.
Yeah, actually, a lot of our mindset portal is to kind of try to be an Intel inside to other people's machines and systems. Because it's so simple, we try to lean into that and integrate into other systems. So for one of our pharma partners, we've already integrated into their robotics. We work with people like Fresenius Kabi and Thermo Fisher, integrating with their clinical scale machines. That's more relevant for cell therapy, but it's a similar mindset of just be the Intel inside to these other systems. We call it powered by portal, but that's kind of the mindset.
And you have put some considerable effort into working with T cells. As we're coming back now to the cell therapy, what exactly do you do and why is this important for cell therapy?
I think cell therapy right now went through a phase of everyone was really excited. And then now it's probably going through a phase of everyone's not so excited because it kind of ended up being a 1 or 2 trick pony. I mean, it can kill certain blood cancers, but it tends to really struggle elsewhere. And these delivery problems are a good illustration of that issue. So for example, if you want to go after a solid tumor of sorts, the types of mechanisms you would want to engineer there are avenue of targeting, which is called TCR-based targeting, as opposed to CAR. So most blood cancer-based stuff, people use CARs, but there's another mechanism you would prefer to use in solid tumors, which is TCRs. The problem with TCR-based T cells, the way they currently make them, is that because of the manufacturing process and the way they expand them, et cetera, these things are crap. They are terrible phenotypes. They're exhausted. They can't do anything. Poses a problem because if you have a bunch of dysfunctional T cells that you put into the body, they're not going to do anything. And an example of what we've shown we can do is we can deliver RNA that codes for 2 very potent cytokines called IL-2 and IL-12. These things are like cocaine for these cells. They really jazz them up and get them going. And now you can really get a lot of function out of them. But the problem with giving these cells IL-2 and 12 is that this stuff is very potent. And currently all you can do is inject IL-2 and 12 into the whole body. And so your whole system goes crazy and the toxicity is unacceptable. But in our case, because we can deliver RNA that only allows your T cell of interest to express its own IL-2 and 12 and not flood the body with it, now you can get much more potency out of them. There's a lot of other esoteric immunology mechanisms I can get into where this is very enabling, but that's just one example of you go from crappy T cells to much more functional T cells, and that alone can be a huge difference maker for hitting solid tumors.
And that wraps up part 1 of our conversation with Armon Sharei. We've covered the fundamentals of intracellular delivery and gotten a glimpse into the future of cell therapy manufacturing. Join us next time for part 2, where we will explore drug screening applications and dive deeper into the technology that could transform patient care. Hey CTOs, are your cell therapy manufacturing challenges keeping you up at night? I specialize in optimizing bioprocesses and scaling production processes. Let's tackle those unsolvable problems together. Book a free strategy call with me at Brühlmann Consulting, and thank you so much for tuning in. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.